Bacteria are host to a special group of viruses called bacteriophage or phage. Phage go through multiple rounds of infectious life cycles with bacterial cells. In this experiment, we will study the virulent phage T2 by using techniques to observe phage activity indirectly through their impact on bacterial host cells. Like many viruses, phage are too small in size to easily visualize, so we can exploit the fact that they multiply only within living cells to observe their activities indirectly. One phage particle introduced into a layer of dividing bacteria will produce a zone of lysis in the opaque film of bacterial growth. This zone of lysis is called a plaque. An infected host cell bursts, or lyses, and liberates dozens of new phage particles which then infect neighboring cells. When handled properly, each phage particle produces one plaque. Any material containing phage can be titrated through dilutions, and the plaque count can be used to estimate phage concentration, analogous to the colony count for bacteria. Before we infect any bacterial cells, we must first titrate the phage, forming a serial dilution of the original stock of T2 bacteriophage. We will be using a 1 in 10 dilution, a total reaction of 1 milliliter, and perform 7 dilutions in total. Dividing 1 milliliter or 1000 microliters by the dilution factor of 10, we arrive at 0.1 milliliters. This means we'll be adding 0.1 milliliters or 100 microliters of the phage sample into 0.9 mil or 900 microliters of the diluent. This will be repeated across 7 dilutions. Dispense 0.9 ml of phage diluent into 7 tubes and make 10-fold serial dilutions of the phage 10 to the negative 1 to 10 to the negative 7. Remember to mix thoroughly at each step before moving on to the next dilution. Select the layer agar from the heating block at 52 degrees Celsius. Add 0.1 ml of the E. coli culture, and then add 0.1 ml of a diluted phage sample, 10 to the negative 1 to start with. This provides the phage with bacterial host cells to infect, as well as nutrient agar for the bacteria to grow. Mix the tube gently, pour its contents onto an empty agar plate. Make sure the solution is spread evenly across the plate and allow it to solidify at room temperature. Repeat this process for each of the phage dilutions. Add 0.1 ml of the diluted phage, 0.1 ml of E. coli culture, mix, then pour onto an empty agar plate. All of these plates will be incubated at 37 degrees Celsius overnight. After incubation at 37 degrees Celsius, you can see the plaque-forming units, or PFUs, present on the plates, representing where phage have infected and lysed the bacterial host cells. This is an indirect measure of phage activity, but you can visualize the change in PFUs throughout the series of phage dilutions. If we multiply the number of PFUs on the plates with discrete countable plaques by the corresponding dilution factor, we can calculate the concentration of the original bacteriophage stock as PFU per mil. For example, if we detect 10 PFUs on the 10 to the negative 3 plate, we can divide 10 PFUs by the dilution factor of 10 to the negative 3 to arrive at 10,000 PFUs in the 0.1 mil of phage. If we express this as PFU per mil, this figure then becomes 100,000 PFU per mil in the original bacteriophage stock. We can perform this calculation multiple times using plates with different dilution factors of phage and come up with an accurate estimate for the original phage concentration. Using this indirect method of observing phage activity through bacterial host cell lysis, we can estimate phage concentration without ever visualizing the phage particles themselves.